so showcasing the potential that these isolates were resistant to erythromycin as well as clindamycin. And again, this rate hasn't changed much over the years, but it does seem that it always stands approximately 25 to 30% if we look at individual years. And we had so far, out of all of this collection, only one isolate with potential putative BBP mutations leading to reduced susceptibility. And we're looking into testing this isolate in more details in the laboratory, as well as some of the other mutations that we identified in this collection. If we look at the specific proteins present on our disease population, again, as expected, we do see the rib protein dominating the data set, in particular when it comes to neonatal infections, as it is primarily associated with serotype 3. If we look at the adult populations, again, alpha protein seems to be more dominant because it's more associated with serotype 1A, and as well can be found sometimes in other serotypes. But overall, which is good news, of course, if we look at all 4 proteins present in our isolates, we do have nearly 98% of them present in the disease isolates. In particular, it's good news for late and early onset, which would be closer to nearly 100% coverage if the vaccine is brought in. If we look individually, again, if in particular age groups and the only single protein presence in each cases, the presence there again is a bit more diverse, and is again associated with that genetic clonal features of the GBS, where specific surface proteins are more likely to be identified in specific serotypes. So for example, here the table illustrates that serotype 3 is almost exclusively showcasing the rib protein presence, and as well having that hypervirulent gene HVGA, which we know again that has been reported before, that these are some of the key virulence factors as well present in the serotype 3, primarily dominated by CC17. If we look at 1A, as I mentioned, ALK1 seems to be dominated, but you do see the other proteins from alpha family, like protein family, also present. Serotype 5 is dominated by ALK2/3, and as well serotype 2 is being alpha. So we can see that as well when it comes to serotype 5 and 2, we see a bit of more diversity of which proteins has been present, and that can be linked in back to the actual clonal complex of the isolate rather than the serotype alone. But if we look overall at the 6 key serotypes that are included in the polysaccharide vaccine, they would be very broadly covered by the protein vaccine as well. So next, what we did, we were very curious to see if there's actually any protein allelic variants present when we look at these targeted proteins. And we did identify that, though at very low numbers, we did find some variants when it comes to alpha protein. So in this case, we found 5 variants. The substitutions usually affecting the D1 and D2 regions of the protein, and some of mutations have actually led to a positive to negative or negative to positive charge change within protein. But again, as I said, even though we detected 5 variants, there were actually at very low numbers, usually just one isolate having this particular allelic variant. If we looked at the rib protein, which is of course as the most common and within our data set, we were very curious to see what we find. So we found more variations, which again is expected. If you have more isolates, more diversity, you might find that. And primarily the identified substitutions were affecting the D1 part of the protein, again, some leading to potential charge, but it didn't seem yet at the moment that any of those mutations would lead to particular change in how the protein is binding to some of the immune systems. So again, it's good news. And again, even though 16 variants were reported, these, again, only 1 maximum to 3 isolates that had some of these variants present. And the same with ALK1 protein, that we found 7 variants within our data set with substitutions affecting D and 1. So it really seems like if the mutations are happening, it's like the 2 regions are the most common ones. And we are investigating further to see actually what is the predicted mechanism for each of these allelic variants that we have identified from other proteins as well, not only the rib that we have looked at the moment. So just to summarize, we all know now that Rib gene is associated primarily with the dominant serotype in the neonatal cases, serotype 3 clonal complex 17, which was the dominant not only in the LODs but in LODs in the UK data set, as well as we know it is globally dominating. And that particular genes that code for those proteins that are targeted by the protein vaccine, the presence or distribution of them varied between the different age groups as well as different serotypes, depending if you were looking at one of the proteins only or if you're looking at the combination of proteins present in the particular isolate. But overall presence of these proteins was close to I said like 96 to 99, nearly 100%, which is promising when we think about the vaccines. And of course, we are carrying on further phylogenetic as well as modeling work to understand better the potential impact that those vaccines might have on the disease-causing isolates, as well as looking into some of the carriage isolates within the maternal and neonatal cohorts, but also that the adult cohorts and the carriage of GBS in the gut. So I'm working on this project with my collaborators in UK Health Security Agency and as well my colleagues in the Imperial College. And I just want to thank you all for coming here to ISSAD. As you can see, these are pictures from the previous ISSAD, so it's really nice to see how this conference is growing each time it's happening. And I really look forward to talking to you over the coffee breaks and all the rest of the conference. Thank you.
Thank you, Elita. So we should definitely take a picture like this at the end, Claudia. So now we are moving from the UK to Portugal. And our next speaker is Elizabeth Martins.
The most recent epidemiological study of the Streptococcus agalactiae causing neonatal disease in Portugal in the period of 2016 to 2025, where I'm going to tell you about the emergence of a macrolide resistant and a multi-drug resistant lineages. I forgot to tell you, I'm from the Faculty of Medicine in Lisbon, Portugal. So I dare not to make a long background statement about GBS disease, because I'm sure you all know, and maybe some of you more than I do, but I'm telling you about neonatal disease, which mostly divides into the early onset within the first week of life and late onset up to 2 to 3 months. So in Portugal, we have since 2004 the first recommendation for screening of pregnant women that was not a health directive, but it was a recommendation of the neonatology section of the Portuguese Society of Pediatrics that recommended the screening of pregnant women and the prevention of perinatal disease. It was only in 2011 that we had an official norm issued by the public health authorities, which established, similarly as to what was being done in the United States, the screening of pregnant women at 35 to 37 weeks gestation, and the recommendation of intrapartum antimicrobial prophylaxis for pregnant women carrying GBS. And there was a subsequent study based on voluntary reporting of GBS disease that showed a reduction in the incidence of disease of nearly 40%, as well as the case fatality rates, similarly to what was observed in many other countries. So the study I'm telling you about is based on a laboratory-based surveillance program, which is a Portuguese group for the study of streptococcal infections, in which, in this case, it included 24 Portuguese hospital centers throughout the country that were asked to submit to a central laboratory ours all GBS isolates recovered from in cases of invasive disease in neonates. And so we progressed to the characterization of these isolates mostly documenting the changes in the serotypes, clonal composition, and antimicrobial susceptibility. So we have a small number of isolates, 162 in this period, similarly distributed in early onset and late onset disease, and mostly recovered from blood, but also from cerebrospinal fluid and peritoneal fluid. And we performed the whole genome